Hey there, and welcome. Have you ever thought about the fact that inside every single living thing there's a code? A really complex, beautiful set of instructions that literally writes the story of life itself. And today, we're going to learn how to read it. It kind of seems like magic, doesn't it? The idea that a single drop of blood or maybe a tiny strand of hair can unravel a whole mystery, identify a person, and tell their story with almost perfect accuracy. So, how is that even possible? Well, to figure it out, we've got to start with a little mystery of our own. Okay, so this is our mystery. It's an evidence table from a criminal investigation. We've got samples from a victim, a couple of suspects, and the crime scene itself. In a bit, we're going to see a unique genetic fingerprint for each of these, a pattern that looks a lot like a barcode. Stick with me, because by the end of this, you'll know exactly how to read these patterns and crack the case.
All right, first things first. This whole idea of a genetic fingerprint. We need to get a handle on what this signature actually is. It's a pattern that's just as unique to you as your actual fingerprint. A biological signature that, with very, very few exceptions, belongs to one person and one person alone.
All right, so to really crack this code, we've got to zoom in. I mean way in, all the way down to the molecular level inside our cells. That's where we're going to find the molecules holding this incredible blueprint. The stars of our show are these things called nucleic acids. You've definitely heard of them, DNA and RNA. These are the master molecules, the absolute keepers of the genetic code that gets passed down from generation to generation. Both DNA and RNA are written in this kind of four-letter alphabet made of chemical bases. Now, three of those letters are exactly the same: adenine, guanine, and cytosine. But that fourth letter, that's a huge difference. DNA uses a base called thymine, while RNA swaps it out for a very similar one called uracil. It seems like a small swap, right? But believe me, it has huge consequences for how they work. And this table just lays it all out perfectly. DNA is a double-stranded helix, it uses a specific sugar, and it's got thymine. The easiest way to think about it is like this. DNA is the master blueprint. It's super protected, very stable, and locked away safely in the cell's nucleus. RNA, though, that's more like a quick photocopy. It's single-stranded, uses a different sugar, and has uracil. It's the temporary working copy that can leave the office to get the job done. Okay, so if DNA is the permanent, precious, master blueprint, what happens when a cell needs to divide? I mean, you can't just give the blueprint to one of the new cells and leave the other one hanging, right? You have to make a perfect copy, and that process is called replication. And it's a really beautiful and elegant process. First, the DNA double helix unwinds itself. Then, just like a zipper, the two strands break apart and it unzips right down the middle. Now, each of those original strands becomes a perfect template. Then all these free-floating building blocks come rushing in and form a new matching strand for each half. And the result? Two perfect, identical DNA molecules. And this right here is the secret to its accuracy. By using one old strand to build a new one, it's like having a perfect guide every single time. It ensures an incredibly precise copy. It's just a flawless system for passing on those genetic instructions every time a cell divides. So we've got the code, and we know how to copy it. But a blueprint just sitting in a drawer is, well, it's useless, right? So the next step is to actually turn that code into a creation, to build the proteins that do all the work in our cells. It all kicks off in the nucleus, in a process called transcription. See, the cell is super protective of that master DNA blueprint. It doesn't want to risk it. So instead, it makes a temporary copy of just one specific gene. And that copy is our friend RNA, specifically messenger RNA, or mRNA. It's basically a short message copied right from the DNA. So once that mRNA message is made, it gets to leave the safety of the nucleus and head out into the main part of the cell, the cytoplasm. There, it finds this little cellular machine called a ribosome, and this is where the real action happens. This is translation. The best way to picture this is like a construction site. The mRNA is the set of instructions. The ribosome is the factory that reads the instructions. The amino acids are the raw building materials, and then there's another type of RNA called transfer RNA, or tRNA, that acts like a delivery truck, reading the code and bringing the exact right amino acid to the factory floor. So you can just imagine this ribosome chugging along the mRNA track, while the tRNA trucks keep dropping off the right amino acids in the perfect order. The ribosome then links them all together into this long chain. But it's not done. That chain then folds up into a really specific, complex 3D shape, and that is what makes a working protein that can do its job.
Okay, amazing. So we've gone from the tiny blueprint all the way to the finished product. We've seen how DNA gets copied and how its code is used to build the proteins that make us who we are. So now it's time to zoom all the way back out and solve that crime we started with.
And here we are, back at the scene of the crime, or at least the evidence table. Now that we understand how the code works, we're ready to see the results. So after all that, what is the secret to actually reading these things? The secret? It's all in the sequence. You see, while most of our DNA is identical to everyone else, certain parts of our DNA have these little repeating patterns, almost like a genetic stutter. And the length of those repeating sections is totally unique to you. The lab technique sorts these DNA pieces by their size, and that's what creates that unique pattern of bars, your personal genetic barcode. Okay, so now all we have to do is play a game of spot the difference. We just match the patterns. Take a look at the sample from crime scene 2. Now look at the suspects. See it? The pattern of bars is an exact match for the profile of suspect X. Suspect Y's profile is totally different. The code doesn't lie. We know suspect X was at that crime scene.
And you know, this isn't just cool stuff for crime shows. This powerful tech is used every single day. It's used to identify criminals, sure, but also to figure out paternity, to connect long-lost relatives, and even to identify victims of disasters. It really gives a voice to those who can't speak for themselves.
And this is the one single idea that makes all of this possible. Your genetic sequence, all 3 billion letters of it, is uniquely yours. Unless you have an identical twin, that DNA profile is the ultimate identifier. It's even more reliable than a fingerprint.
So we've seen this incredible, elegant code that builds life itself. It's a nearly perfect system. But it makes you wonder, right? What happens when it's not perfect? What happens when there's a typo, a little glitch in the code? Well, that's a story for another time.